Welcome, everybody. My name is Sharon Rounds. I'm the PI for Advanced Rhode Island CTR, which is an NIGMS-funded center to support the development of research infrastructure in the state of Rhode Island. And we have a monthly research seminar featuring distinguished investigators and awardees from that Advanced CTR was pleased to be able to support. So today we're very pleased that we have two of our grant resubmission awardees sharing the seminar time today. Both Dr. Freiman from Brown University and Dr. Manuel from the University of Rhode Island received grant resubmission awards and were very successful in their NIH submissions. So we're very proud of their accomplishments. The first talk today will be given by Dr. Richard Freiman, who is professor and vice chair of the Molecular Biology, Cell Biology, and Biochemistry Department at Brown. And he'll be discussing his work on developmental aspects of female and male reproductive longevity. And I would just note, I would appreciate it if people could hold their questions for the end of the talk and just put them in the chat, and we'll have an opportunity to entertain them. So take it away, Rich.
Okay, thank you, Sharon. And thank you for all coming today and listening to me. And I really appreciate the chance to talk about some of the research that's going on in our lab. And as you mentioned, funding for this grant resubmission award came sort of in between grants when the lab had very few resources. And we were very appreciative and still are very appreciative of the support that we got. And I hope that program continues for a long time. So today I'm going to talk about female and male reproductive longevity, and having a hard time. Let's see. Let me try something. All right. So to get started, basically it's pretty clear that reproductive lifespan or longevity differs between males and females. And this is basically looking at relative fertility rate. And you can see that relative fertility rate declines in females before it declines in males. And that overall, the statement that female fertility is more strongly influenced by age is sort of very apparent to all of us. But I would like to talk about sort of a comparison of male and female reproductive longevity from a developmental biology point of view. And the take-home message of hopefully this talk today will be that some of the origins of either decreased or pathological reproductive longevity probably arise during development. And while many of us are thinking about fertility in our 30s and our 40s, and many of the clinicians that are trying to manage fertility issues in couples basically don't really, I don't think most of us, even the scientists, including me, don't think about early development as the place where some fertility problems may arise in your life. So if we look a little bit more closer to this, we all know, and we've seen this kind of graph for throughout our careers, but on the left is the incidence of trisomy, and on the x-axis is maternal age, and you can see that in the late 30s or so, the incidence of trisomy increases dramatically in women. And that is sort of a clear sign that there's this sort of age-associated decline in oocyte quality. What's a little bit less known is that there is also a paternal age effect on gametes. This is a study now published a while ago, and it was looking at the potential of paternal age or older fathers giving rise to individuals with autism and schizophrenia at higher percentages. And that they found a correlation in this study of that. But for here, what's important is what's graphed here is the number of de novo mutations and the father's age on the x-axis. And you can see that as the father ages, the number of de novo mutations in the genome of the sperm that the father is delivering for conception is increasing. And I think this paper concluded that there are 2 additional mutations every year of life. So there, in addition to female oocyte decreased gamete quality, there's also decreased gamete quality in men. And this is probably important because basically, both the sperm and egg are contributing to the genome of the child. So I'm not really going to talk about any of this sort of adult stuff today. And most of what I'm going to talk about today is sort of happening in the sort of early development or mid-gestation development. And this is a very complicated slide, so it would take me probably a half hour to go through this if I went through everything, but I'm just going to summarize some of this. This is a timeline of mouse and human development from fertilization through puberty, okay? And basically, it's focused on looking at the development of the germ cells and the gonads. And in both species, basically early development of the gonad and early development of the germline have many similarities. So again, on the top is the mouse here, and on the bottom is the human. But I'll talk a little bit more about the mouse because this is the species that we study in the lab. Upon fertilization, you have a zygote here when the sperm nucleus fuses with the egg nucleus, and that is basically the beginning of development where you have cleavage divisions, formation of blastocysts here, and then there is basically specification or an induction event sort of at around embryonic day 6.5 or 6.75, where you have these cells in the posterior of the embryo being instructed to become what are the first germ cells. And these first germ cells are called primordial germ cells. And this is happening very, very early in development. So this is just after implantation and after gastrulation. So you have the birth of these primordial germ cells, and similar specification is happening during human development. And then these germ cells, that initial pool of PGCs is very small, but they start proliferating, and they're highly mitotic, and they also are migratory, so they migrate through the hindgut, and then they colonize the gonad at about mid-gestation and about E12.5. And it's about this time when colonization of the gonad is happening that the somatic gonad is being instructed to form an XX gonad or an XY gonad. And in the sex chromosomes, when you have 2 X chromosomes, the somatic cells are often instructed to become supporting cells of the ovary. And in an XY gonad, the supporting somatic cells become the Sertoli cells, and those Sertoli cells support spermatogenesis. Okay, and then there is a time point sort of during development where it's really not understood what's happening, and that's sort of boxed here in red. And this is the time point in development that my lab studies both germ cell development in the ovary, in the embryonic ovary, and the embryonic testis. Okay, so there's a lot known about the PGC specification and the early development and sex determination, the early development of the gonad. And there's also a lot known about the adult function and the adult ovary and testis, where you have, after puberty, you have ovulation that leads to the formation of the female gamete, and ejaculation leads to the production of the male gamete, sperm. But there is this sort of time point in the middle here where there's a lot going on. So just to look a little bit more carefully at this, this is sort of in the mouse, which is on the top here. This is sort of mid to late gestation, and it's sort of right around the middle of development and the time of birth. Basically, these PGCs in the female gonad are instructed to become oocytes, and they start the process of meiosis. So the process of meiosis in females starts very, very early. And what I highlighted down here is that this process is likely happening in the human ovary at between 8 and 20 weeks of gestation. So you already have oocytes going into meiosis. And then in the case of the mouse, these primordial follicles that form, these primordial follicles are the ovarian reserve. And this is a finite resource. The adult ovary or the postnatal ovary doesn't have a mechanism of making new oocytes. So women are born with a finite pool of primordial follicles or oocytes, and that number declines throughout their lifetime. So at the same time that that's happening, and these are important events in female fertility, there are events happening in the embryonic testis where these prospermatogonia, so these are now male germ cells, they're going through a period of quiescence, and that period of quiescence is associated with them sort of becoming adult or spermatogonia right at the time of birth or after birth here. So you have this sort of development of these sort of transitory embryonic male germ cells that will become spermatogonia and spermatogonial stem cells in the male. So the male mammals have a spermatogonial stem cell. So unlike females, they have an adult stem cell population that can give rise to new sperm and new male germ cells throughout most of their lifetime. But all of this is happening at the same time, and it's happening in a very sort of sex-specific manner here, where these female germ cells are going into meiosis and then arresting in meiosis, and these male germ cells are just becoming mitotic spermatogonia, and they're not going to go through meiosis until a little bit later in development. So again, many of these hallmarks are conserved in the human ovary here, and the human testis. And this is sort of a time point where we know very, very little about how the germline and how reproductive longevity is going to be affected by the events. And certainly in the human, that's the case. And now we have tools to study this in the mouse, and this is what my lab has done. So in addition to these developmental events, there's also another sort of major epigenetic reprogramming that's happening in these PGCs. So what's actually shown here is the amount of DNA methylation in these primordial germ cells, and at early points in development when they're first specified, and they have high levels of DNA methylation, so they have their their DNAs are modified by cytosine methylation, and there's likely to be histone modifications that are associated with that DNA methylation. But as these PGCs, and this is in both males and females, migrate to and proliferate to the gonad at 12.5, they lose their DNA methylation patterns are erased. Okay, so they're actually, once they get to the gonad, they have very, very low levels of DNA methylation. And now you have this epigenetic reprogramming that comes after this. And this happens a little bit earlier in males than females. These prospermatogonia remethylate their DNA pretty quickly. And by the time of birth, that remethylation is thought to have been largely completed. There's a little bit more of a delay in when the female oocytes remethylate their genome. But this short delay, there is sort of similarity between these two. And in both cases, as a field, we don't really understand the mechanisms of how these germ cells regain these epigenetic modifications. So they're erased and they're reestablished, but how that's happening. And then just shown below here is that these female oocytes are in meiotic arrest, while the male germ cells are in mitotic arrest for this period of this epigenetic reprogramming. But after birth, they start proliferating again. And it's during this period that is thought to sort of give rise to spermatogonia and spermatogonial stem cells that are really, really important for the long-term maintenance of spermatogenesis. So there's a second wave of epigenetic reprogramming that people are much more familiar with, which happens in the embryo when the zygote, the DNA methylation is erased in the genome of the embryo and then is reestablished after in the inner cell mass after formation of the blastocyst. So I brought up this sort of part because this is less well known, but it's clearly important if these cells are going to become the precursors of all of the gametes that that an individual is going to make in their lifetime. So in my lab, we study a transcription factor called TAF4B, and TAF4B is part of a TF2D complex. So there's a canonical form of the TF2D complex that contains TBP and the TATA box binding protein that binds to the TATA box in the core promoter regions of many different promoters. And this canonical pre-initiation complex regulates global programs of gene expression. So TF2D itself does not really have anything to do with fertility. It really has to do with maintaining levels of transcription of many, many genes in the genome. But there is a version of TF2D that we study, or we think we study, that has this additional subunit in it, and this additional subunit is called TAF4B, and this subunit is promoting oogenesis and spermatogenesis in the mouse that we study, but it's likely to be conserved, and I'll show you some data for that later in the talk in human fertility. So we studied these TAF4B knockout mice for a long time, and about 10 to 15 years ago, they sort of became clear that they were kind of an unexpected model of premature reproductive aging. So what do I mean by that? Well, it turns out that the ovary of TAF4B deficient mice, that ovary and those female mice are completely infertile. They have an accelerated loss of ovarian follicles. So this is a ovarian follicle that you would find in a healthy control mouse, and you can see that in the TAF4B mice, they basically have very unhealthy pycnotic granulosa cells, and the granulosa cells don't adhere to the oocyte like they do in the healthy ovary. So you have this accelerated loss of ovarian follicles, and this mouse models primary ovarian insufficiency, which is a sort of a clinical term for infertility that affects about 1% of women worldwide. So this is a major cause of infertility in our population. Um, the the phenotype of the male TAF4B knockout mice is a little bit different. They're initially fertile. They produce a couple of litters, but then they stop producing litters after the first or second litter. They have a very early germ cell loss and testicular atrophy, and that's shown here. Basically, you can see these empty tubules, and these are the adult knockout mice, while their control het mice or wild type mice have these beautiful seminiferous tubules that are producing sperm, and these are fertile mice. So this mouse in our hands models non-obstructive azoospermia, or NOA, that is also a cause of a high percentage of infertility in humans, in men. So we wanted to know what was going on. Where were the defects of these mice? Were they adult onset defects, or were they happening earlier? And it took our lab a long time, but we finally traced back their deficiency of germ cell loss to the perinatal period. So either late embryogenesis or early postnatal life. So in the perinatal ovary, Kate Grive, when she was in the lab, found that that ovaries, TAF4B deficient ovaries, had a much reduced ovarian reserve, even at postnatal day 1. And then Lindsay Lovasco, when she was in the lab, showed that even in the male, there was a reduction in embryonic germ cells at late embryogenesis at E18.5. So while we initially started thinking that we were studying an adult onset phenotype, where we would sort of be looking at fertility problems in mice that were happening when they were adults or maybe adolescents or something like that, it turned out to be, to our surprise, that these events where these mice were actually losing their germ cells were happening during late embryogenesis and early postnatal life. So it starts to sort of to make sense to us when we looked at the expression of TAF4B in embryonic germ cells. And you can see in blue here, this is the expression of TAF4B, and this is the mRNA level of TAF4B. And this level, TAF4B, goes up at embryonic day E14.5, and that is the same in the males. There's an increase in TAF4B. So there's very, very low levels of TAF4B in germ cells before this sort of mid-gestation time point, and then you have a large amount of TAF4B being expressed, and it's probably during these next few embryonic days that TAF4B's function, or its first function in these germ cells, is really important. This marker in gold here is just the expression of polo-like kinase 1, or PLK1, and this is decreasing as TAF4B levels increase. And in females, this increase is happening when mitotic PGCs are transitioning to meiotic oogonia, and in males, those mitotic PGCs are transitioning to become quiescent spermatogonia. So in both cases, in male and female germ cell development, you have this sort of a loss of cycling, or this sort of exit from the cell cycle that leads to different developmental outcomes. So where we are today in this type of analysis is that In the female gonads, we looked a little bit more carefully, and this is the work of Kimberly Apt, and she looked at embryonic day 18.5, and this is a large list of oocyte differentiation genes, and this is our TAF4B deficient germ cells, and these are our het and our wild type, and these females are both fertile. And you can see that there's a reduction here in these heat maps of these oocyte differentiation genes. So one of the things that these oocytes get stuck at is at this time when they should be expressing this large battery of genes to go on to further differentiate into healthy oocytes, they fail to do that. They also fail to downregulate a lot of these meiosis 1 genes that are listed here. So you have this upregulation of meiotic genes here because that these oocytes fail to decrease the expression of those meiotic genes that they're trying to turn off at this time point. So one of these oocyte differentiation, Kate Grives Labs works on at Brown, and that's called UCHL1. And you can see that there are high levels of UCHL1 in the TAF4B wild type E18 point ovary, but there's much less staining for UCHL1, this critical oocyte differentiation protein. And this likely sort of that and other gene deficiencies leads to the demise of this ovarian reserve. So what I've now tried to show you is that we had these phenotypes in the adult ovary. So this is a 10-week-old ovary. This is a wild type and a knockout. And you can see the knockout has many fewer follicles and is smaller than the wild type ovary. And this ovary is not producing any functional oocytes. But the timing of when the defect of this infertility is happening way back here at E18.5. So this is an example where something is happening at E18.5, and it's those regulatory events are really important for the long-term maintenance of healthy oocytes in adults. So what is TAF4B doing in the embryonic testis? And again, we started looking at these prospermatogonia. We've been sort of running this project simultaneously in the lab, and it's not common for labs to study both male and female reproduction in the same lab, but Miles Bartholomew has been working on this project in my lab. And what Miles discovered was that at E18.5, there is a unique subpopulation of embryonic male germ cells that arises that isn't present at E15.5 or at E16.5. So this is in wild type embryonic OC4 GFP plus spermatogonia. When we look at our mutants, we actually see the absence of that side population here. So the wild type and hets have this side population, and the mutants in red here are lacking that side population. And we're now thinking that that side population may provide some cells that actually promote the healthy development of the spermatogonial stem cell pool as it develops a little bit later in development. So what I've showed you today is basically how TAF4B has these early functions to support reproductive longevity in both the male and female mice. So in oogenesis, it's required for establishing the ovarian reserve and early meiotic progression. In spermatogenesis, it's required for the development of this prospermatogonia that will give rise to a spermatogonial stem cell pool soon after. But does any of this have relevance in humans? And it turns out this isn't our work, but this is work published in the field. And this is a human pedigree here showing that there are these 4 brothers. 3 of these brothers have homozygous mutations in TAF4B that causes NOA, and one of these brothers is oligospermic, so has subfertility. And so there are a couple of more brothers and a couple of sisters here. We don't really know if these sisters were infertile, but we would predict from this type of pedigree that if they were homozygous mutant for TAF4B, having this sort of same truncating mutation, they too would be infertile. So when we're thinking about fertility, we need to be thinking about both males and females at the same time. And I was scratching my head thinking it's kind of strange for a lab to be working on fertility of both sexes at the same time, since this is never done before. But others are thinking about this too. And this is just a paper that came out in 2025 on a DNA repair gene called MCM8. And in that family, they see gonadal dysgenesis in both genders. And the brother that has this homozygous mutation has NOA, and the sister that has this homozygous mutation has POI. And in both the TAF4B family and the MCM8 family, I would predict that there's early demise of the germline very, very early in development. So I think it's important to really think about reproductive longevity, thinking about both sexes, but also thinking about when the timing of the cellular or molecular defects are actually having their most important effect. And with that, I'll basically thank Kim Seymour, Miles, and Kimberly Apt. They did most of the work that I was able to show today. And I appreciate all the funding, especially from the advanced CTR. And I'll take any questions. Thank you.
Okay, great. That was a remarkably succinct description of a very complicated process in both sexes. Thank you. That's great.
Thank you.
We are a little short on time, but I will take this opportunity to ask, is it now in the clinical realm, are people who go to fertility clinics being screened for these mutations?
Not yet, but it's soon. So, and I think in the last couple of years, there's a very renewed interest in reproductive longevity, just in general, especially in women. And at these conferences that I've been going to recently, there happens to be a lot of clinicians that are sort of bringing their work into similar realms. And I think, some of them have been studying Turner syndrome women, and they have a very, very low ovarian reserve. And again, it's likely that those women are having, early signs of fertility that we haven't been looking for clinically. And it's not necessarily easy to measure the ovarian reserve in young children. So that's something that labs here at Brown are working on. But it's
Fantastic. But so, yeah, I could imagine that if one came up with that sort of a mutation, it might change the direction of the workup rather considerably. So, okay, I think we're going to have to move on to our next speaker.
okay.
Thank you very, very much. And let's see, can we change the slides here?
Stop sharing.
Stop sharing, yeah. Good. And can we bring up Dr. Manuel's slides?
Yep, give me a second.
So
Okay.
our next speaker is Marin Manuel, who is an assistant professor in the Department of Biomedical and Pharmaceutical Sciences in the College of Pharmacy at the University of Rhode Island. And he also was awarded a grant resubmission award. And so he'll be telling us today about his work on the transcriptomic profile of functionally identified mouse spinal motoneurons. Take it away.
All right. Thank you so much, Dr. Rounds. Thank you, everybody, for being here. And so, yeah, I'm going to present this project that was funded by RI-CTR, and I'm very grateful for their help in trying to get this this project started. So you'll see that the title is a bit aspirational because we don't really have the transcriptomic profile of functionally identified mouse spinal motoneurons yet, but that's where we want to get to. So before I get started, I just want to point out that really this is the most important slide. These are the people who are actually doing the work in my lab. I have been at URI for a little bit less than 5 years now, and we've started a great collaboration with Dr. Quinlan, and so we are like working very closely together. And so this is our current lab. Today, I'm mostly going to present some work that's being done by Rebecca here in the front. Okay, so this project started quite a while ago when I tried to submit a grant to NIH to study, as the title says, the transcriptomic profile of motoneurons. And this grant was initially, this was an R21 grant, so an exploratory grant. And so this was not funded, but there was some good comments on it, and we wanted to resubmit it. But for that, we needed some more preliminary data to include in this grant. And so that's where RI-CTR came into play. I got this award, and this allowed me to perform some pilot experiments and to include in the resubmission of the grants that I did at the end of last year. Unfortunately, that grant was not funded. Again, they had some issues, and I'll discuss this with you maybe later. And so this is where we are now, and I'm hoping to be able to resubmit this grant at the beginning of the next calendar year. And hopefully this one will be third time the charm, as we say. Okay, so my lab is mostly interested in spinal motoneurons, which, in case you don't know, are cells that are located in your spinal cord or your brainstem, and have send their axon outside of the central nervous system to form synapses with muscle fibers. And so they are the cells that control muscle contraction. Right? And but if you look at a textbook, so I took this image from the Candle textbook, you see that motoneurons are just these cells, like this homogeneous population of cells that innervate the muscle fibers. But the thing that we're mostly interested in the lab, or have been interested in for many years now, is that in fact these motoneurons are not a homogeneous population, and there are many types of motoneurons. In fact, in this review that we published a while back, we've identified 7 different types of motoneuron, some of which don't even produce muscle contraction at all, and they innervate these special organs that we have in our muscles that are actually sensory organs and not motor organs. But for the purpose of today, we're going to focus on these alpha motoneurons here, which are written here with this Greek letter alpha. These are the motoneurons that we think about when we talk about motoneurons. These are the cells that innervate the muscle fibers that generate contraction in our muscles. And so even in these alpha motoneurons, we have at least 3 types of motoneurons based on the muscle fiber type that they innervate. Right? And so we have these in motoneurons in blue here innervates what's called type 1 muscle fibers, whereas the FR type innervates type 2A muscle fibers, and the FF innervates type 2B muscle fibers. And so studying this diversity of motoneurons is extremely interesting and very relevant to pathology, because in many pathologies, in particular amyotrophic lateral sclerosis, which is the pathology that I've been studying the most, these motoneurons degenerate, but not randomly, and they degenerate in a specific order, where the FF motoneurons are more sensitive than the FR, and the S motoneurons are the last to generate. And so one of my goals is to try to understand what are the things that make these motoneurons more susceptible to the disease, or these motoneurons more resistant. And so for this, you need a way to identify the type of motoneuron and to be able to recognize the kind of motoneurons that you're dealing with, so that you can study this population differently from this population. And so to do this in a mouse model, we developed a technique that allows us to record the motoneurons in vivo inside the mouse with a microelectrode that we drive inside the spinal cord, and we can record the motoneuron intracellularly inside the spinal cord. But then we perform these experiments in vivo in a anesthetized but non-paralyzed animal, so that when we inject currents into the motoneuron, here this is the current that we inject through this microelectrode here, we elicit an action potential, right? So these are the electrical messages that are passed between neurons, and these are the messages that are traveling alongside the nerve, down the nerve of the motoneuron up to the synapse between the motoneuron and the muscle fibers, and then activate the muscle fibers. And so we can, in the same preparation, we can inject this current, trigger an action potential in the cell body that will travel down the axon, activate the muscle fibers. This is the electrical signal that's generated by the muscle fiber. It's called a motor unit action potential. And then this action potential will trigger the contraction of the muscle fibers, and we can record the force that's generated by these particular muscle fibers activated by this single action potential here by attaching a force transducer to the tendon of the muscle. Okay, so on paper or on the screen here, it looks kind of pretty. In practice, it's a bit more messy. So this is the kind of setup that we use to do this recording. We have a mouse here that's deeply anesthetized, right? It's like if you were to go to the OR, it doesn't feel anything. It's placed under artificial ventilation, and then we do a bunch of surgery to expose the spinal cord. Thankfully for you, it's hidden behind this post here. We expose the muscle over here. We're working on the calf muscle of the hind limb. And then we have this electrode here. This is an intracellular glass microelectrode that we drive inside the spinal cord to record the motoneurons inside the spinal cord. And then every time the motoneuron that we're recording from is firing, we observe a muscle contraction at the periphery here that can be recorded with this force transducer. And so using this technique, we are able to identify the kind of motoneuron that we're recording from. Right? So every time we generate an action potential, we have a muscle contraction. This is called a twitch contraction, individual contraction. And by looking at the shape of that contraction, we can identify whether the motoneuron that we're recording from contracts, or the motor unit that we're recording from, I should say, contracts slowly, like this one, for example, or contracts quickly, like these two, right? And so based on this, we can separate the S-type motoneurons from the F-type motoneuron. And then we can also try to fatigue the muscle fibers, which is another characteristics of the muscle fibers, is that some muscle fibers tend to fatigue more than others. And so you can see that here we do like a series of contraction for a long period of time. And we see that the S motor units do not fatigue. The force remains constant throughout, whereas the FF motor unit fatigue, and so their force decreases over time. So using this series of criteria, we are able, for the first time in mice, to recognize the type of motoneurons that we're recording from, and to try to study what's going on specifically in those units, or in those units that makes those more resistant or more vulnerable, and those more resistant to a disease condition. Okay, so I don't know if I got this point across, but these are very complicated experiments. It's quite tedious, and we can only look at one motoneuron at a time, right? And we characterize all of the properties of this motoneuron, but it is very challenging to do these experiments. And so it would be great and to have a way to access directly those motoneurons on a histological slice of spinal cord, for example. Right? The problem is that when you look at a histological slice of a spinal cord, you cannot differentiate the different kinds of motoneurons. So if you're not familiar, this is a cross-section of a spinal cord. We have the central canal over here. We have the dorsal horn over here, the ventral horn over here. And in magenta here, we labeled the motoneurons. And what you can see is that the motoneurons have different sizes. We have some tiny motoneuron and some big motoneurons. The tiny motoneurons correspond to the gamma motoneurons, which we're not going to talk about today. The big cells here correspond to the alpha motoneurons. But among these big cells, it is impossible at this stage to differentiate whether this particular motoneuron is an S or an FR or an FF motoneuron. And so whether this neuron is going to be more resistant or more susceptible to the disease. And so that was one of the goal of my lab, was to try to find ways to to classify these different motoneurons so that we can recognize them and study them in more details. So this is not a new idea, and several labs have tried to propose different markers that we could use to identify different population of motoneurons. Here recently we published a paper trying to summarize all of these different markers, and you can see that some markers have been proposed that would be able to differentiate between those different kinds of motoneurons. The problem is that these markers are based on basically very subtle differences in sizes between the FF and the S motoneurons, and they haven't been validated in a physiological condition with a physiological readout of the force generated by these motoneurons. And so this is something that we've done, and we tried one of those markers, right? We started with this marker here called chondrolectin. And we've done the full experiment where we've recorded the motoneuron during an experiment. We've characterized the force profile of that motor unit so we could classify it in one of the three categories. And then we inject a fluorescent dye inside the cell body of the motoneuron. So now, among all of the motoneurons of the body of the mouse, we have one motoneuron that we recorded during the experiment where we know everything about this motoneuron, and that motoneuron is fluorescent. And so then we can find that motoneuron post hoc after the experiment on histological slices. And then Rebecca performed in situ hybridization to look for RNA for that particular gene called chondrolectin. And what we found is that there are two populations of motoneurons that do not express chondrolectin, or very low levels of chondrolectin, and motoneurons that express very high level of chondrolectin. And so chondrolectin was proposed as a marker for fast motoneuron. And so people would say, oh, this motoneuron that expresses chondrolectin is a fast motoneuron, an FF or an FR, whereas this one is a slow. But in fact, what we found is that chondrolectin is not a marker of fast versus slow, but rather a marker of big versus small. So in this plot here, we have the amplitude of the twitch, so the amount of force that's being generated by the motoneuron. And what we found is that chondrolectin is expressed only in the cells that generate lots of force, but not in the cells that express low amount of force. And the slow motoneurons are located in this quadrant over here. So what we show is that chondrolectin is a marker of size rather than a marker of speed. And that, yes, motoneurons that express chondrolectin are fast, but not all fast motoneurons express chondrolectin. So just to show that there are still much more work to do, and we have to validate those markers that were proposed during the by other labs. The problem is that, again, this work is very long, very tedious. It took us years to get these motoneurons here, and so we wanted to find a faster approach. And At the same time, you know that there was this explosion in the approach of single-cell transcriptomic. And so a lot of lab tried to study the transcriptomic profile of motoneurons. So here is one such study. I should have put the reference here. But they did bulk sequencing of single cell of all the cells in the spinal cord, including the motoneurons. And they get these kinds of plots here where each dot represents one cell. And you can see here in this particular cell, in this particular plot, each dot represents one motoneuron. And you can see that you have different clusters, but how do these clusters relate to the physiological type that I mentioned earlier, the slow, the FF, and the FR, is an open question. We don't know which cluster corresponds to which, or even if each of those types correspond to individual clusters in this approach. So our project, and the one that I submitted to NIH, was to flip the script and start from the physiology and to go to transcriptomic instead. And so the goal was to identify the type of motoneurons that were recorded during in vivo experiments, and then perform transcriptomic on those individual motoneurons to try to identify individual markers or unique markers that could be used to identify these different population. And so for this, I propose to perform transcriptomic analysis of functionally identified motoneurons using laser capture microdissection, and then to validate those markers using multiplex fluorescence in situ hybridization. So the workflow would be to perform these in vivo experiments that I mentioned before, right? Similarly to what we've done before, we study a particular motoneuron, we characterize the force produced by its motor unit, and then we inject a fluorescent dye inside the cell body of that motoneuron. But now, instead of looking at only one marker by in situ hybridization, we are putting this spinal cord under a laser capture microdissection microscope, so that we can actually dissect the cell body of that motoneuron, collect it in a tube, and perform single cell sequencing on that particular motoneuron, and then compare the expression patterns of genes in motoneurons that we recorded and we characterized as either S, FR, or FF, and then validate this by in situ hybridization. So this is the project that I submitted to NIH, and what they said is that this looks very nice, but you haven't proven that you're able to do this step here. And so this is where advanced CTR came into play and allowed me to perform pilot experiments to show that this step here was working correctly. And so for doing this, I tried to simplify the experiment a little bit. Instead of doing the full recording in vivo and recording only one motoneuron at a time, I thought we could prove that we can microdissect motoneurons and study their transcriptome by doing an injection of a fluorescent tracer inside the muscle here. So we inject this tracer called CTB. It's the beta subunit of the cholera toxin, coupled to a fluorescent molecule. So we inject it in the muscle. This molecule is taken up by the axons of the motoneuron and transported all the way to the spinal cord, where the cell body of the motoneurons will then become fluorescent. The cell body of the motoneurons that innervate this muscle become fluorescent. And so doing this, we are able to then get a bunch of motoneurons that are fluorescent. We don't know the type of these motoneurons that will come later, but at least we have now fluorescent motoneurons that innervate this particular muscle. And then we went to the microscope, the laser capture microdissection microscope, and we see these cell body under the microscope. And then we can use our laser to cut the cell body of those motoneurons and collect the cell body of the motoneurons. And what we've done is this pilot study where we try to prove that we could get good enough RNA from one particular cell to perform sequencing on. And so I did this test study where we collected either one cell or two cells or 3 cells in one well. And we had a total of 18 samples, and we'd send them to be processed and sequenced by an external company called Admira Health. And so this was the part that cost a lot of money, and where most of the money that Advanced CTR was kind enough to give me went in. And so the result of this is that we were able to show that even with 1 cell in a well, we are able to get a good enough RNA that we can get a good sequencing. And we see that yes, having more cells help, but not that much. And in most cases, 1 cell gives us enough RNA that we can perform a high number of reads. We don't have that much contamination by ribosomal RNA. And so I believe we are in a good position to do the RNA sequencing on identified motoneuron. This is a principal component analysis of the genes, showing that the cells that have a lot of reads here tend to cluster together, and which again point out to the good quality of the RNA. So I was pretty happy with this result, and so I resubmitted the grant to the NIH. Oh yeah, and we've also at the same time developed a multiplex fluorescence in situ hybridization process, where instead of looking at 1 single gene like we were doing previously with chondrolectin, now we can look at least 4 different genes expressed by the motoneurons. And so that will speed up the validation step much, well, at least 4-fold or 3 or 4-fold. So this was also some new development that was financed by Advanced CTR. So happy with all of this. We resubmitted the R21. Unfortunately, it was still not funded, and there was still some criticism that were raised, like the fact that yes, we have the number of reads, but we didn't provide the number of genes that were detectable. And so that's something that some analysis that I need to perform again on the samples that we have. Some criticism that were pretty much, in my mind, unfounded, but clearly showed that I needed to explain this better in the grant. The fact that the iontophoretic dye injection, for example, was not demonstrated, even though we had these experiments here that were done with the same technique where we injected the dye in the motoneuron. So these are things that I need to explain a little more. And yeah, so I think those criticisms are fairly easy to respond to, and so I'm hoping that we'll be able to resubmit this grant and be successful in getting this work funded. And I'm going to stop here. Thank you so much.
Fantastic. Thank you very much. And you know, it's really interesting to see, you know, I think that the way you use this funding to do those expensive analyses is exactly the type of purpose for the grant resubmission awards. So good luck on the next go round, and
thank you so
very
much.
exciting work. I think we're kind of at time now, so I'm not seeing anything in the chat. So I think at this point, what we'll do is thank both Rich and Marin for really interesting discussions. We're good luck to both of you and this very interesting and important work. We next up in November, Suzanne de Lamont, who is in the pathology department at Brown, will be discussing her work on alcohol related brain damage. So with that, I'll thank everybody and have a great day. Enjoy the upcoming 3-day weekend. Bye-bye.
Thank you so much.